Hi, my name is Maggie Brisbane, and I'm currently a postdoc at the Woods Hole Oceanographic Institution, working in the labs of both Harriet Alexander and Max Saito. Today I'll tell you about one of my projects looking at whether Phaeocystis colonies host consistent microbial assemblages, and how those microbial assemblages might affect Phaeocystis growth. Phaeocystis is a globally abundant haptophyte phytoplankton that can form massive blooms in many different regions across the globe. Here, I'm showing the relative abundance of different Phaeocystis species based on the 18S rRNA gene sequences in the UKBank database. These data demonstrate that different Phaeocystis species are dominant at different latitudes, with Phaeocystis poucheti clearly most abundant in the Arctic, Phaeocystis antarctica most abundant in the Antarctic, and Phaeocystis globosa generally most abundant in temperate regions and across the equator, but there are a few other species in these regions as well. Phaeocystis poucheti, antarctica, and globosa, however, are the three species that are most well known due to the size of their blooms and their unique ability to form large colonies. These three species have bimorphic life cycles and transition between free-living flagellate cells and large mucilaginous colonies with thousands of cells embedded in the colonial matrix. As you might imagine, the production of these colonies has a significant ecosystem effect, and they are accessible to different grazers compared to the flagellate cells, among many other reasons. In the wild, Phaeocystis blooms often precede, follow, or replace diatom blooms. For example, in the Southern Ocean, Phaeocystis antarctica blooms often occur before diatom blooms, whereas in the Southern Gulf of Maine, Phaeocystis poucheti blooms are replacing diatom blooms. Several studies have already shown that there are complex and specific interactions between diatoms and bacteria that can give diatoms a competitive advantage in some situations. But so far, interactions like this have not been demonstrated for Phaeocystis. And this is despite the fact that the Phaeocystis colony represents a distinct and potentially ideal interface for bacterial interactions, partly because it is large and sticky, making it a good attachment site, but also because it is full of complex carbohydrates and organic nitrogen that can be food sources for bacteria, and Phaeocystis is a copious producer of DMSP, which can be a bacterial attractant, and lastly because the colony contains acrylate, which might selectively deter some bacteria. Therefore, I have two main research questions for this project and this talk. First, do co-cultured bacteria influence Phaeocystis growth? And secondly, do Phaeocystis colonies harbor consistent bacterial communities? And within this, are colony-associated bacterial communities similar across strains cultured from different places and at different times? And can colonies re-recruit similar bacterial communities from seawater after their own bacterial communities are removed through antibiotic treatment? So to answer these questions, I worked with four strains of Phaeocystis globosa, three that I acquired from the NCMA culture bank, the CCMP1528, CCMP629, and CCMP2710, and one that I isolated from the Kuroshio current more recently. And in addition to representing a large geographic range, these four strains have been in culture for different amounts of time. So CCMP 629 was originally isolated in 1982, 1528 was isolated in 1992, 2710 was isolated in 2006, and then I isolated the Kuroshio strain in 2019. And using these four strains, I first rendered all four of them azenic, so I treated them with an antibiotic cocktail, and then I checked them with HECT fluorescent DNA strain, and I often did transfers of the cultures into marine broth to see if there was any bacterial growth. And then I generated growth curves for both the azenic and the control cultures of the four Phaeocystis globosa strains under nitrogen limiting and vitamin limiting conditions. And I evaluated the bacterial communities that were associated with individual colonies and that were free living in the media for each strain using 16S rRNA gene sequencing or metabarcode analysis. And lastly, I exposed each of the azenic strains to free living bacteria in natural coastal seawater, and then I evaluated the bacterial communities that were recruited to the colonies and to the media. So first, let's look at the results from growing the azenic and the control cultures in media that didn't have any additional B vitamins. So on this plot, there are 5 culture cycles. So 1, 2, 3, 4, 5, and then each box represents one strain. And the antibiotic-treated cultures are in black with the solid circles, and the untreated controls have the dashed lines with the open triangles. And we can see that over time, the antibiotic-treated cultures stopped growing, whereas the untreated controls continued to grow despite the fact that there was no additional B vitamins provided to them. So this is an indication that the bacterial communities in these cultures are actually providing an essential B vitamin to the Phaeocystis. Conversely, if we look at what happens when we withhold nitrate, the same exact plot, so 5 culture cycles, antibiotic-treated cultures are in black with the solid circles and black lines, and the untreated controls are open triangles with dotted lines. So over time, the untreated controls stopped growing, and the antibiotic-treated cultures actually kept growing despite the fact that there wasn't additional nitrate being added into the media. So this is an indication that the bacterial communities in these cultures are competing with the Phaeocystis for nitrogen resources. Next, let's look at the results from the 16S metabarcoding analysis. So first, looking at the alpha diversity and the different sample types, the colonies, so there is 3 to 5 individual colonies per culture replicate, and there was 3 culture replicates. So in each box, we have 9 to 15 replicates. And in the media, there's 3 replicates for each, which represents the bulk of the whole culture being filtered down and then having the DNA extracted from the filters. So we can see right away that the individual colonies have less diverse microbial communities associated with them. And for the original communities, it's about 1/3 as diverse. And in the recruited communities, there's about half as many ASVs in each of the colony samples compared to the media samples. So essentially, it looks like the colonies have a subset of the available bacteria in the culture media that are associated with the colony surface. So looking a little bit deeper into this data, this is a principal coordinate analysis of the Bray-Curtis distances between the communities. So on the bottom left here, we can say that the bacterial communities in the natural seawater were very different than in the cultures, regardless of strain or whether it was a colony or just the culture media. And then the square boxes represent the culture media, and we can see that the recruited culture media is more similar to the natural seawater than the original culture media, and that both the culture media from the original cultures and from the recruited cultures are distinct from the colonies, but the colonial microbial communities actually converge and become quite similar. So there's a winnowing down of this very diverse community in the seawater. Some of that diversity gets recruited into the media, but not all of it. And then some of the media diversity gets recruited onto the colonies, but not all of it. And once we get down to those that remain on the colonies, it's actually quite similar in the recruited and in the original communities. And we can look a little bit deeper at these results. So this plot, I know, looks absolutely crazy at first glance, but I'm going to walk you through it. So each box represents a sample type. So this top box is the original colonies, and then the next box is the recruited colonies, and then below that we have the original media, and below that we have recruited media, and at the very bottom we have the seawater. And each column represents a single ASV, and they are grouped by their taxonomy based on a FAST tree at the top here, and then they're colored by family. And then the colors in the bubbles are the strain of Phaeocystis, and the size of the bubble represents the relative abundance of that individual ASV. And what you can see pretty quickly is that there's some columns here that are very consistent. So it's a single ASV that is present in all of these different samples. So we can see that there are some ASVs that are very abundant in all of the colony samples, whether it's recruited or original. So for example, the Alteromodalis are very abundant in the original and the recruited colonies. Some other ones that are abundant in both include Burkholdeриalis and the Rhizobialis. And I think that Rhizobialis is actually maybe the most interesting of those that are abundant in the colonies, because this is a bacteria that's also found to be associated with, or a bacterial group. There's a lot of diverse bacteria within the group, but within this group there's a lot of bacteria that are associated with plant roots and that have been proven to provide nitrogen, fixed nitrogen, and also some plant hormones that encourage growth. So this is going to be a very interesting group to look into in the future. There's also some other interesting patterns that I'd like to point out in this plot. So I highlighted 3 of them here. So in this first case, we have some ASVs that are present in the seawater and that are recruited into the media, but they are not recruited into the colonies. They are recruited into the colonies, but they are not present at all in any of the original samples. And in the second example, we have some ASVs that are present in the seawater, and they are recruited into the media and not into the colonies. And then in the last example that I think is an interesting pattern, we have some ASVs that are present in the seawater, and they're recruited into the media and into the colonies, and they were also abundant in the original samples in the colonies and in the media. So we have a couple different patterns that are showing up with the ASVs, where some ASVs are being recruited to just the media, some ASVs are being recruited to the media and into the seawater, and some of those were already present in the original communities and some of them were not. And I just want to point out that this plot only has the ASVs that are present in at least one of the Phaeocystis samples. So there were a whole lot more ASVs that were in the seawater, but this plot is really just focusing on those that were in the Phaeocystis communities. But just to really drive home how diverse the bacterial communities in the seawater was, compared to what was recruited to the Phaeocystis media and colonies, this tree shows all of the ASVs that were in the data set. And if they are highlighted in blue, they were only found in seawater and they were never recruited into any of the samples. And there are some whole clades here that are only found in seawater and are never recruited. So for example, the SAR 406 is only in the seawater and never recruited into any of the Phaeocystis samples. So a couple conclusions from this research so far. Bacterial communities associated with Phaeocystis globosa colonies are a consistent subset of the total community that lives in the culture media. The bacterial communities that are associated with Phaeocystis globosa are consistent across strains, despite these strains originating from very different locations and having been isolated decades apart. The colonies of the different Phaeocystis globosa strains are able to re-recruit similar bacterial communities from natural marine bacterial communities, and they're similar to each other, and they're similar to their original communities that they were hosting. And then finally, these bacterial communities, in addition to being consistent and reproducible, they are also having an actual effect on Phaeocystis growth. So they can either promote or inhibit Phaeocystis growth, depending on depending on the environmental conditions. So they're either providing a or competing for an essential nutrient. So I think that there's going to be a lot of follow-up to this work looking for the specific strains that are contributing these different functions, and I'm really excited to keep working on this project. And with that, I'd like to thank you for listening. and thank my advisors, Harriet Alexander and Max Saito, and my thesis advisor, actually, Satoshi Mitrai, and also Don Moran, who has helped a lot with this research. And you can contact me by email or find me on Twitter if you have any questions. Thank you very much.